Hi everyone, I'm Keith Page, Director of Scientific Development at Asteran Bioscience. In this webinar I'll be discussing an approach that we've developed that aims to refine and accelerate the progression of immunohistochemical assays through the IHC assay validation process. Immunohistochemical assays are increasingly used to detect and quantify protein biomarkers in tissue sections. And for these assays to be used to their maximal utility, assay development and validation are of paramount importance. Assay validation follows guidelines set out in the Validation of Analytical Procedures document, and it usually requires quantitation. And while quantitative immunohistochemistry is regularly performed, relative measures, optical density and others, are frequently used. The case study I'll be presenting builds on what is run in quantitative IHC and takes us towards a prototype immunohistochemical standard curve, converting classical brown staining into more quantifiably meaningful data. First, a bit of background about Asteran Bioscience. In 1996, two former GSK scientists, Gordon Baxter and Bob Coleman, had the notion that discovering drugs for therapeutic application to humans might be accelerated by performing as much preclinical research as possible using human tissues. This led to the formation of Pharmagene, which combined its own drug discovery activities with a classical fee-for-service contract research model. The USP of Pharmagene was the use of human tissues for human drug discovery. A few years later in 2000, Randall Charlton founded a human tissue biodepository in Detroit, which provided human tissues and crucially the accompanying clinical data to researchers in industry and academia. Pharmagene and Asteran merged in 2006, were acquired by StemGen in 2012, divested StemGen in 2014 and rebranded as Asteran Bioscience in 2014, which is where you find us today. Over the years, we've grown in experience and developed expertise in deriving high-value data from this most challenging, but ultimately the most valuable research biomaterial, human tissue samples. In our UK facility based in Royston, about 15 miles south of Cambridge, we offer a range of research services. Gene expression profiling, quantifying mRNA expression in normal and diseased human tissues using TaqMan real-time RT-PCR. Fluorescent in situ hybridization, identifying the genomic status of oncology samples using standard and custom-made FISH probes. mRNA in situ hybridization, using fully automated branch DNA technologies from Affymetrix and advanced cell diagnostics. Some of you may have heard about our evaluation of these competing technologies in a webinar developed by my colleague Paul Murdoch. Immunohistochemistry for target validation, single and multiplex localization, development of assays for using companion diagnostics, which you'll be hearing about today, and tissue cross-reactivity, including the evaluation of a wide range of antibody-like molecules. Lead optimization work based on use of cell-based assays, including our proprietary culture system OrganDot, where we use human islets and cancer cells to more closely replicate elements of diabetes and cancer for compound evaluation. So this is not the most wildly exciting slide in the deck perhaps, but these two regulatory accreditations from the UK Human Tissue Authority and the MHRA were very challenging to obtain and underline our rigorous adherence to all components of the Human Tissue Act, particularly in that all our human tissue samples have been obtained under conditions of informed consent with approval for research use. And the certificate from the MHRA underlines our attention to scientific and QA detail in delivering our tissue cross-reactivity service to the standards required for GLP. So today's webinar is going to look at a precise topic. In focusing on the immunohistochemical assay development and companion diagnostics, I'll be drawing on our capabilities in IHC and tissue arrays, but also in quantitative image analysis, cell culture, measurement of RNA expression and RNA knockdown technologies. Asteran runs a state-of-the-art molecular pathology laboratory. The staff who work here are fully trained, experienced scientists, including 4 GLP study directors, and from time to time we draw on the expertise of consultant clinical and veterinary pathologists. As you would expect from a GLP lab, we use documented SOPs from every step, from tissue dissection and storage to running our assays to data analysis and the delivery of reports. Our immunohistochemical workhorse platform is the DACO autostainer. We have 3 of these, IQ, OQ and PQ validated, but we also now routinely work with 2 Ventana platforms, the Discovery and the Ultra, and the Lycobond platform where appropriate. All our slides from chromogenic assays that can include immunohistochemistry and in situ hybridization are scanned using a PerioScanscope and the data made available to our clients via secure online telepathology. The bulk of our IHC works falls into target validation using antibodies to identify precisely where and at what level in normal diseased human tissues the target proteins are expressed. Quite often we're asked to develop assays to novel targets using untested antibodies. Anybody who regularly evaluates human tissue using IHC will know only too well perhaps that the quality of commercially available antibodies is variable, to say the least. For this reason, we invest significant effort in identifying specific antibodies and the optimum conditions for their use. Working with paraffin-embedded sections, for example, depending on the platform, will assess multiple antibodies across multiple antigen retrieval conditions, across multiple antibody concentrations, alongside appropriate and rigorous positive and negative controls. Control cell lines and tissues are helpful in these stage 1 optimization assays and in selecting controlled tissues, where we have access to a proprietary database of over 2,500 mRNA expression profiles across 72 pathologically normal human tissues. So in the absence of definitive protein expression data, this mRNA expression resource can be particularly helpful. Once we've completed the stage 1 assay development, we're confident in applying that assay to the stage 2 tissue set. So we've semi-formalized our IHC assay validation process using the term 7 steps. So we'll be looking at the practical application of these steps in subsequent parts of the webinar, but to cover these briefly, step 1 relates to the assay design and the inclusion of the most rigorous and appropriate negative controls. Step 2 looks at the data. Are we seeing specific staining in the expected cells and the expected cellular compartments? Step 3 evaluates the antibodies in Western blot. Are we detecting proteins of the expected molecular weight with a margin for post-translational modifications? Step 4, do control cell lines that show the expected binary data, either positive or negative? Step 5, does more than one antibody generate qualitatively similar data? Step 6, how similar are your data to those in the published domain, with the very important caveat that not all antibodies have the same quality, and hence not all data has the same quality either. Lastly, step 7, RNA knockdown experiments with immunohistochemical determination of protein levels. So excluding step 7, RNA knockdown, these steps can combine to establish a case for whether an antibody is specific. Steps 1 to 6 individually, none of them are definitive and shouldn't be taken as such. So moving on to the case study, we're going to be looking at validation arrays and development of companion diagnostic immunohistochemical assays. So the assay development usually follows these guidelines, and these are taken from the ICH guideline document. The specificity component we address quite thoroughly using the 7 steps approach, but the accuracy, precision, and particularly the quantitative elements present more of a challenge to conventional IHC, where qualitative endpoints are more typically used. So in our case study, we're looking at the development of an IHC assay to MET, high affinity tyrosine kinase receptor to HGF, hepatocyte growth factor. We selected this target because of its importance in a range of human cancers and crucially because of the availability of the tools that we needed. So in developing the case study, we selected 4 different cell lines with known levels of MET expression. We acquired a full-length recombinant human protein to MET, and there are lots and lots of antibodies to MET. We've just selected a few, and we're going to be showing data from 2. Both antibodies are reported by the manufacturers to be suitable for use in paraffin-embedded sections of human tissues. So a quick note on image analysis. In order to quantify the staining intensities in this study, we used digital whole slide scanning and an unbiased measurement system, which to my mind quite closely replicates the process a pathologist would use in assigning 0 to 3+ scores to stained sections. The NSR value is a simple algorithm within ImageQuant, which is part of the Aperio package of image analysis applications. And what it returns when you ask it to quantify DAB staining is a relative proportional score. When the system assesses staining intensity, it breaks the range of intensities down into 4 bins. Negative, no staining, weakly stained pixels, positive pixels, and strong positive pixels. NSR is the proportion of strong positive pixels relative to all the measured pixels. So when NSR is 1, all the pixels the instrument has measured fall within the strong positive bin. And so we use this method to quantify chromogenic staining in the 3 components of the immunohistochemical validation assay. Here's a scan of an H&E stained section from the validation array, and it shows quite nicely its organization. There are 4 rows of samples, cell cores, tissue cores, and 2 rows of spiked homogenate cores. The top row of the 4 cell lines, the relative abundance of MET in these lines is quite well documented, but briefly, SNU5 is a primary gastric cell line with a very abundant MET expression. HT29 is a colorectal cancer cell line with known moderate levels of MET. MB231 is a breast cancer line with low expression of MET. And crucially, SNU1 is a gastric metastatic cell line where MET is known to be absent. Under the cell lines, we have tissue cores from 5 human gastric carcinomas. These are included to provide information on the cellular and subcellular localization of the target protein. For MET, we're expecting to see expression concentrating at the membranes of cancer cells. Below the cancer cores are the spiked homogenates. These are duplicate cores of a non-human tissue matrix spiked with full-length recombinant human MET over a known concentration range. In this case, it goes from 20 down to 0.01 micrograms per mil. The first experimental evaluation of these 2 antibodies was in Western blot, where lysates from all 4 cell lines were run out, transferred and probed with both antibodies. Let's look first at gel A. This was probed with D1C1. Lane 1 is the recombinant MET, and we're picking up 3 main bands, consistent with alpha, beta and precursor MET. The intensity of the bands, particularly those around 150 kilodaltons, matches what we'd expect given the known measurement of MET in the cell lines. In gel B, the other antibody, EP1454Y, was used, and while superficially the gel looks cleaner, there is reduced and incomplete detection of the recombinant protein and a similar level of band intensities across all the cell line lysates, which is unexpected given that what we know about the variability of MET expression across these cell lines. The histogram on the right shows data from the immunohistochemical analysis of the cell lines in the validation array, the NSR values for each cell line probed with both antibodies. And there again, there appears to be a difference in the staining intensities across each of the cell lines for the 2 antibodies tested. The inset data shows the negative control, the rabin IgG, the maximum intensity of which is superimposed on the main histogram, and you'll see it's at a very low level, such a level which doesn't interfere the interpretation of the specific staining. So there appears to be differences in how these 2 antibodies perform in Western blots in cell line IHC, with D1C2 more closely reflecting the known relative abundance of MET. So let's have a look at how these antibodies perform over a standard range of immunohistochemical conditions, in fact the ones recommended by the manufacturers on the data sheets. For both you can see an appreciable concentration effect, and the panel to the right shows the impact of varying antigen retrieval pH. We tested standard pH 6 and pH 9 on the autostainers in this particular experiment. So we're using heat-mediated retrievals. The increase in staining intensity with antigen retrieval at pH 9 is quite typical, but with some antibodies, particularly rabbit polyclonals, we could keep a close eye on the level of non-specific staining in the negative controls. This slide shows how the antibodies perform in spiked homogenates, highlighted in the array scan bottom right, where the red box outlines the 2 rows of spiked homogeneous samples. This data shows a clear relationship between the NSR value and the MET concentration for both antibodies. Data resulting from D1C2, the blue bars, reduces to background, the green bars at 2.5 micrograms per mil, while data from EP1454 never decreases to background levels, suggesting some degree of non-specific staining, data consistent with the cell lines and the lysates and the Western blots, in fact. The right-hand graph recasts the same data on a log scale where the different behavior of the 2 antibodies is more immediately appreciable. This combination of image analysis with an IHC standard curves allows quantitative indices of the assay to be measured. So for example, over the concentration range where MET was detectable by D1C2 under these assay conditions across 20 down to 2.5 micrograms per mil in the recombinant samples, there's a linear relationship between NSR and MET concentration. We are also able to add values to important assay performance criteria, detection limit, quantitation limit, linearity and range. For the evaluation of accuracy, under optimal assay conditions using the gastric cancer cores, we assessed 3 MET antibodies, D1C2 and EP1454, and a new one, 8F11 from Novocastra. This time we're looking carefully at cellular and subcellular expression. D1C2 is on the left, EP1454Y in the middle, and 8F11 to the right. Considering 3 main criteria, there appear to be differences in how these 3 antibodies perform. For example, D1C2 is labeling up tumor cells prominently, the mesenchyme is negative, and the membranes are very prominently positive for MET. EP1454Y, tumor cells are positive, but so are large proportions of mesenchymal cells, membranes are positive. 8F11 looks qualitatively quite similar to D1C2, but the membranes you'll notice appear negative in this assay. At higher magnifications, the difference between the immunohistochemical performance under these conditions are clear. The inset shows the non-immune control incubation, and you can see quite nicely that D1C2 labels up cancer cells prominently, but the staining is concentrated at the cell membrane, so you can see that quite nicely. For the ABCAM antibody EP1454Y, you can see cancer cells are also strongly labelled, but mesenchymal cells are also labelled as well. So the next experiment we ran to evaluate the performance of these two MET antibodies is effectively the acid test, specific lentiviral knockdown of MET mRNA in two of the cell lines, the moderate and the low expressing lines. 3 lentiviral particle transfections were tested called MET 1 to 3, and the optimal condition was determined based on measurement of mRNA, MET mRNA. Infections with clones MET 2 and 3 were found to maximally reduce MET mRNA expression by over 90% actually, so both cell lines were accessed using immunohistochemistry. These scans show MET immunoactivity in the HT29 cells treated with negative control lentiviral transfections, the top 3 scans, so HT29 medium, non-target and no insert, so it's a medium expressor for MET, and the non-targeting and the no insert lentiviral treatments have no impact, as you'd expect, on the measurement of MET by D1C2. When you have a look at the 2 effective lentiviral transfections, MET2 and MET3, you can see an over 90% knockdown in the number of cells expressing MET, which is prominent and very striking and not unexpected. You're not really expecting to see 100% disappearance in a target protein, I don't think. So D1C2 is once again supported. The specificity of this antibody is well supported by the lentiviral knockdown data. How does the ABCAM antibody perform in the same assay? Again, the 3 negative controls have no impact on staining intensity. When you have a look at the 2 conditions which knock down MET, you see no real impact on the expression of MET immunoreactivity in this moderately expressing cell line. Again, which is consistent with the Western blot, consistent with the cell lines, and consistent with the spike recombinant system as well. So where are we? We've assembled a strong case for the use of D1C2 in further companion diagnostic assay evaluations based on Western's cell line IHC, tissue IHC, and quantitative IHC using calls from spiked homogenates, and crucially in the data you've just seen from the RNA knockdown assays. All this data points to D1C2 being specific and use of EPY of 1454 under conditions tested, resulting in data with a significant non-specific component. So based on the specificity data, we selected D1C2 for further ICH assay validation. So how does D1C2 perform across the remaining assays required for thorough validation? We have a few more to look at here. The first one we're going to look at is assay repeatability. And what this assay does is it looks at the consistency between slides within a particular assay. All of our DACO autostainers are IQ, OQ, PQ validated, which measures the same endpoint, so we're not expecting too many surprises here, I don't think. And representative data from one cell line, the SNU5, and one of the gastric cancer cores shows how the NSR value varies between slides in a single assay. So the upper data set shows the cell lines, and the histogram to the right shows hardly any variability between staining intensity across the replicates tested, and this is probably what you'd expect to see from a cell line. It's a homogeneous sample and it's approaching the ceiling of 1 for the NSR value. Variability was more pronounced with the tissue samples across the same assay, and that's probably reflective of the increased sample complexity and heterogeneity. To assess intermediate precision, we used sections for the validation assay and D1C2 under optimum conditions and ran 9 replicate assays using 3 different DACO autostainers, which for reasons of scientific rigor we refer to as snap, crackle and pop. And as expected, variation in the assays was low for all samples assessed. The scans show 2 cell lines, SNU, 5 HT29, 1 representative cancer core, and the spike tissue homogenate at the highest concentration for the recombinant MET. And you can see there's no discernible difference really between the assays run on each of the 3 autostainers we have. Run the test. These data were quantified, but I haven't shown you the data here. We next looked at how reproducible the assay was between 2 different laboratories. So working with DACO Cytomation, they generously provided the platform and facility to test our assay under their conditions in their facility. And again, you can see using 3 cell lines, SNU1, HT29, SNU5, representative gastric carcinoma sample and a spiked sample, there's no real difference. And we can look at the data with image analysis to support any between laboratory variability. In fact, when you have a look at higher magnification, you can see that the assay is labeling up cancer cells prominently at the cell surface for both our assay and the decocytomation assay. And then when we're having a look at the quantitative image analysis measuring NSR across some of the samples we used, we can demonstrate more quantitatively that the assays are performing in a manner consistent with accurate reproducibility. So the last data slide shows the assessment of assay robustness. There are a very wide range of assay parameters within immunohistochemistry that we could deliberately change. But in this case, we varied primary antibody incubation time. And this had a clear impact, particularly in evident between 1, 4, and 16 hours at 4 degrees in the intermediate cell line HT29. The staining intensity resulting from incubation with non-immune IgG was minimal over the same range. To summarize then, we have developed an approach combining 3 linked elements: validation arrays, use of spiked homogenate samples, and quantitative image analysis, which can be used effectively to accelerate the progression of IHC through assay validation. Use of spiked homogenates refines the degree to which immunohistochemical data can be quantified by providing a prototype standard curve to a degree consistent with other more traditionally quantitative assays. Taken together, this sort of approach may be of interest to laboratories developing immunohistochemical companion diagnostics. Finally, I'd like to acknowledge and thank my Astrand colleagues who are responsible for generating much of the data that you've seen. Jessica made the array and ran the immunohistochemistry experiments. Mozan prepared the spiked homogenates. Caroline and Sam were responsible for the cell culture and the RNA knockdown experiments. And Howard ran the PCR measurements of MET and cell lines. And lastly, thanks to DACO Pathology Products in Cambridge, with whom we worked to demonstrate assay reproducibility. So thank you for your attention. So there are some questions here which have come through. The first one is, what if cell lines or tissues with known concentrations of the target are not known or are not available? This goes back to something I mentioned earlier on in the presentation. We have a database in-house where we've measured, we've used TACMAN RT-PCR to quantify the expression of mRNA in a very large number of different human tissue samples. And this facility gives us the resource to predict with a degree of accuracy which human tissues are going to be positive or negative for the expression of a given protein, on the understanding that mRNA expression doesn't always translate. We can correlate our databases with those available online, and we can correlate that with the published data in the literature as well. Second question I've got pertains to how much things cost if we were to run this as a service, which ultimately is our aim, of course. The easy answer is it varies tremendously on how much or how little of these assay types you want to run. Each module that you've seen is between $5,000 and $15,000 to run each, and you would expect to commit to 3 or 4 months to have the work done to completion. Last question here, if we're interested in working with you on this type of project, what do we need to provide? For example, antibodies, that sort of thing. Of course, antibodies, if you can provide control cell lines, both positive and negative, ideally over a range of levels of expression, that's helpful. If you can provide us with full-length recombinant protein, or if it's partial recombinant, we need to know exactly its characteristics. And if you can provide us with other information, that's also extremely helpful. We can, of course, buy these as well, but it's easier if you provide us with well-characterized materials from your own lab. Those are the 3 questions to answer. So thank you very much indeed for your time. I hope that was of some interest.